Okay, so assignments have been extended, work on a WebAssign, turn in question checks, work together. I think a lot of y'all are already working together, so keep doing that. And so let's go ahead and look at blood. So blood has, what's it made out of? It's 55% plasma, and the rest are red blood cells. So plasma proteins, water, other solutes. So that's the composition of plasma. Okay, we got another person coming in. Good morning. So we're back on blood. So we got another person. Admit. Okay, we're still presenting. Yep, you're presenting, we're recording. Okay, get rid of that. Okay, back to blood. So plasma proteins are albumins. It's a very high concentration. So that maintains osmotic pressure. So those albumin proteins stay in the blood, and you have globulins, which include hormones and transport of ions, lipids, and immune function. It's very, very important now, what we're talking about. So fibrinogens for clotting, hormones, enzymes, electrolytes, sodium, potassium, calcium, chlorine, bicarbonate, other compounds that are used, glucose for energy, amino acids, fatty acids used in protein production. So blood is a wonderful, wonderful liquid that does all kind of stuff. Okay, whole blood. So formed elements are the red blood cells and all of the white blood cells. Platelets, whole white blood cells. So there's one type of red blood cell. That's it. It's lost its nucleus. Its only job is to be a sac of hemoglobin to carry oxygen. Then we have neutrophils, eosinophils, basophils, lymphocytes, and monocytes. So she said, never let monkeys eat bananas. So that tells you the relative abundance or the percentage of each of the type of white blood cells. Never let monkeys eat bananas. So that's the relative size. You can see that red blood cells are about 7 to 8 microns, 10 to the minus 6 meters. The monocytes are a little bit bigger. So let's talk about these individual elements. So red blood cells, about 7.2 to 8.4. Okay, hello, hello, hello. Admit. Okay, go back to the PowerPoint. So red blood cells have lost their nucleus. They live about 3 months, and then they die. And they carry hemoglobin, which is very important for carrying oxygen to our cells and carrying some of the CO2 and so on. So we had some questions about this slide. So what happens? So a macrophage, where'd a macrophage go? Monocyte is a macrophage. So white blood cells can eat up consume the red blood cells, and they're made in bone marrow. And so recycling. So the new red blood cells are released from bone marrow. They live about 120 days. old and damaged red blood cells are eaten by macrophage, digested. The iron is saved. It goes back into the bones. The liver gets rid of bilirubin and other proteins that come from the degradation of hemoglobin. So also the bilirubin can be excreted in bile and eliminated. So the protein hemoglobin broken into bilirubin and so on. The kidneys are also involved in eliminating some bilirubin and so on. So the first thing that happens is old or damaged red blood cells are consumed, digested, and broken into amino acids. The iron from heme is recycled back into the bones, and elimination of bilirubin can occur in the kidneys and in the large intestine. So they live about 120 days, and they are very, very important in carrying oxygen. So we have a late bloomer. There we go, late bloomer. Okay, let's keep going. I'm not going to ask you anything about this. Just remember there are stem cells or starting cells that lead to production of red blood cells and white blood cells. This talks about bone marrow producing platelets. We'll talk erythropoietin is a hormone produced in the kidneys that works with stimulating the production of reticulocytes and so on. I'm not going to ask you anything about that, so let's just jump through that. Okay, blood types. There's type A, type B, type AB, type O. So type A blood has surface antigens, has a protein. A protein is an antigen. It generates antibodies, and you can, since there's no anti-B being present is fine. Just the opposite here. So if A were present, it would bind to this type B and clump up the red blood cells, and they wouldn't be able to carry oxygen, and they would possibly form clots with platelets, and then also clog up blood vessels. Type AB blood has both A and B, so you don't want any anti-A or anti-B, and type O doesn't have any antigen, so it's the universal donor. So what happens when you have, say, antigen A and anti-A, you can see they clump up and leads to breakup of the red blood cells. So that's why you don't want to make sure, well, everybody has this type blood. You want to make sure that they're given the blood that's not going to cause clumping and hemolysis. Hemolysis means to break down red blood cells. Okay, so white blood cells, the first most abundant is a neutrophil. These are red blood cells. And then we're going to 741. So let's stop there, go back here, stop this recording, stop recording, stop recording, stop.